La última presentación del mismo corre a cargo del doctor Ronald Zsymusiak, quien cuenta con un doctorado en investigación y actualmente se encuentra adscrito al programa de investigación del Centro de Salud del Sistema de Salud en Los Ángeles, California. También él es profesor adjunto de los departamentos de medicina y de neurobiología en la Escuela de Medicina de la Universidad de California en Los Ángeles, y él ha trabajado intensamente en estudios de privación de sueño, demostrando las consecuencias de esto. Ha trabajado con receptores, con el estudio de receptores a adenosina y las alteraciones a nivel hipotalámico en relación a la privación de sueño, y es precisamente de lo que nos viene a hablar, y le agradecemos mucho su presencia. Dr. Zsymusiak, welcome to Mexico City. Thank you very much for being with us.
Thank you for that introduction, and thanks to the organizers for inviting me. I'm delighted to be here. Also, congratulations to the Society on the landmark event of your 10th biannual Congress. So I'm going to speak mostly about systems and circuits that put the brain to sleep. But by way of introduction and context, I want to talk a little bit about what keeps the brain awake. So there's consensus in the field that generalized EEG and behavioral arousal is maintained by multiple ascending neuronal systems that originate, that is, their cell bodies are located in the rostral brainstem, in the posterior hypothalamus, and lateral hypothalamus, and in the basal forebrain. And these have multiple neurochemical signatures. For instance, in the brainstem, there are the monoaminergic and the cholinergic ascending neuronal systems. There are the hypocretin orexin neurons located in the lateral hypothalamus. They're critically important. And in fact seem to be sort of a master regulator of arousal because they send excitatory projections to monoaminergic and cholinergic systems. And then you have the cholinergic neurons located in the basal forebrain, which project directly to the cortex and the limbic system. Now, all of these, to characterize these systems as arousal systems is an oversimplification. They're involved in specific behaviors, specific aspects of cognition, and sort of specialized aspects of arousal. But again, sort of what we think of as generalized brain activation during waking is really emerges from the collective activity of these multiple neuronal systems. And one of the defining features of each of these systems, as one might expect, is when you look at the neuronal activity in each of these cell groups, they're much more active during waking than they are during non-REM sleep. There's some variance in terms of activity during REM sleep. Some of the neurons are like the orexin and monoaminergic neurons are sort of wake selective, that is, they have low discharge in both non-REM and REM sleep. Cholinergic neurons in the brainstem and the basal forebrain are active during both waking and REM sleep. So one of the challenge for any brain neuronal system that is capable of sort of putting the brain to sleep, facilitating the transition from waking to sleep, is that it must help promote sort of rapid and coordinated suppression of activity in these multiple neuronal systems that are spread throughout the brainstem and the basal forebrain. I'm going to talk about a little bit about how that's achieved. So just like we tend to identify neuronal systems that have waking-related functions based on the evidence that they're more active during waking than sleep, one bit of evidence we look for to identify potential sleep regulatory neuronal systems are neurons that are more active during sleep or active during the transition from wake to sleep than they are during waking. And this is an example of some early recordings that our group did in the cat, one of the first identifications of sleep-active neuronal discharge, located, for the most part, in the hypothalamus, and in the rostral part of the hypothalamus, the preoptic hypothalamus. So I apologize for the neural anatomy that will be in the talk. I'll try to keep it simple and point out some landmarks. So this is a sagittal section through the cat brain, through the hypothalamus. The major landmarks here are the anterior commissure, dorsally, the optic chiasm ventrally. This is the front of the brain, this is the back of the brain. So you can see here, microelectrode passes through this part of the brain. One is able to detect fairly frequently neurons with this sleep-related discharge. This is a polysomnographic and electrophysiological demonstration of what the activity of these neurons can look like. This is sort of a basic rat hypnogram, wake, non-REM sleep. This is the discharge, extracellular recorded discharge of a single neuron. This is a rate histogram. Here's the EEG, the EMG. You can see that this cell reliably discharges more frequently when the rat is asleep versus wake, sleep versus wake. So we take this electrophysiological signature of sleep activity in neurons in this area of the brain, which has been implicated with other kinds of evidence for having a role in sleep control as being potential components of sleep regulatory circuit or circuits. A second way to identify sleep active neuronal populations in the brain is to use the method of CFOS immunohistochemistry. So many of you are probably aware that CFOS is an immediate early gene which is expressed when calcium content in cells is high, and so when cells are active and there's calcium uptake, intercellular calcium release, the CFOS gene is expressed. So you can use the evidence of the immunohistochemical staining of the CFOS protein. That can be taken as evidence that that cell expressing the protein has been relatively active in the hour or two before the animal is sacrificed. So, as was originally done in Cliff Saper's group at Harvard, they looked for patterns of CFOS expression in the hypothalamus in rats that had been predominantly awake versus predominantly asleep in the hour before they were sacrificed, and they were able to identify in this ventral part of the hypothalamus, which they call the ventrolateral preoptic area, or VLPO, sort of cluster of neurons that became activated during sleep but were not activated during waking. In certain situations, this VLPO sleep regulatory area can be defined by this VLPO cluster here at the base of the brain, and then what they refer to as the extended VLPO, which is a sort of a dorsal and medial spray of neurons that appear to be sleep active. Practically speaking, when you do these kinds of studies, you have to count cells in brain sections across different animals, and so you have to use some kind of a guiding or counting box so you can compare comparable areas between animals, and this is one configuration that we and others have used to define this VLPO cluster in the extended VLPO. So you can do electrophysiological experiments, demonstrate that there seems to be at least good anatomical correspondence between these neurons that are sleep active and based on their expression of sleep-related CFOS activity and those that show electrophysiological activation during sleep. This is from a study that we did where we essentially made repeated microelectrode passes through this lateral preoptic area. The locations of each recorded neuron are shown as a little white block, and they're color-coded with the grayscale. The darker the square, the more strongly sleep active the cells were based on their electrophysiological activity. You can see that as you go from the dorsal lateral preoptic area to the VLPO, you see an increased concentration of these sleep-active neurons. Again, this is what these neurons look like when you record from them. Here's the action potential, EMG, EEG, higher discharge during sleep versus wake. Here's REM sleep, and this neuron is actually, discharge is slightly higher during REM sleep than non-REM sleep. So a general feature of these sleep-active neurons in the preoptic area is that they are active during both non-REM and REM sleep compared to waking. And these are mean data from populations of cells. And while there can be individual variability between cells, when you look at a group of them, they tend to have similarly elevated discharge rates during non-REM sleep REM sleep and non-REM sleep compared to waking. A second area of the preoptic region where you find high density concentration of sleep-active neurons is an area referred to as the median preoptic nucleus, abbreviated here MNPO. And this is a demonstration with the CFOS method, where the CFOS is stained as a black precipitate here. And what you can see in the sleeping animal is in the, again, our landmarks, the anterior commissure, the optic chiasm, and this is the third ventricle. Here you see in the midline MNPO, high density of FOS staining. As you go a little bit rostral, the FOS staining forms a little cap on the roof of the third ventricle. There's very little FOS staining in these areas in the awake animal. Just to orient you, the red arrows indicate the location of the VLPO cluster. So the other things that we know about these preoptic sleep-active neurons is that they seem to be predominantly inhibitory in nature. That is, they express markers for the inhibitory amino acid neurotransmitter GABA. So they are GABAergic. The majority of the sleep-related CFOS in the MNPO and the VLPO is contained in GABAergic neurons. Now, the Cliff Sapers group had done a series of studies showing that in the VLPO, the neurons are predominantly projection neurons, and they have predominantly descending projection. They project principally to the histaminergic neurons in the tuberomammillary nucleus of the posterior hypothalamus, but also to monoaminergic neurons in the dorsal raphe and the locus ceruleus. So postdoc in my group a few years back, Zsymusiak asked the question, well, given that we have these sleep-active neurons located in the median preoptic nucleus, are they projection neurons? Where do they project? So he did some studies like this one here, where he would inject an anterograde tracer into the median preoptic nucleus, and then look throughout the brain for evidence of axons that are stained, where the tracer has been taken up by cell bodies here and transported down their axons. So this is a few examples of where he sees anterograde labeling following injection like this. Here in the lateral hypothalamus, in the area where hypocretin immunoreactive neurons, here stained brown, are located. And I don't know how well this is showing up with the light, but the arrows here are pointing to black labeled axon varicosities that are sort of lying adjacent to a hypocretin neuron, and you can see anterograde labeled axons throughout this area of the hypocretin neuronal field. Here in dark field, it's perhaps a little easier to see. You can see labeled axons in the dorsal raphe nucleus, in the adjacent ventrolateral periaqueductal gray, here in the locus ceruleus. So like the VLPO, neurons in the MNPO seem to send descending projections to some of the key hypothalamic and brainstem arousal groups. This is an example now also worked on by Aaron using a retrograde tracer. These red fluorosphere beads placed in the parafornical area. This is the lateral hypothalamus where hypocretin neurons are located. And that yields retrograde labeling of cell bodies in both the median preoptic nucleus and the VLPO. And so what Aaron did was to do these injections in a series of animals and then essentially allow the animals to survive. The tracer is taken up by the cells, and then he would kill different groups of animals after a period of wakefulness versus a period of sleep. And so what you can see here in both the MNPO and the VLPO is that, of course, there are lots of retrogradely labeled axons or cell bodies from this tracer injection, but very, very few of those labeled neurons are expressing TFOS. In contrast, when you sacrifice an animal after a period of sleep, there's a much higher density of neurons that are both expressing sleep-related FOS and show retrograde labeling. So this indicates that this projection from these preoptic nuclei to the hypocretin orexin area is sleep active, at least a component of it is sleep active. This is just another example showing the same kind of data with the same approach with tracer injections placed in the dorsal raphe and the ventrolateral PHG. So this sort of leads to the what now is getting close to being dogma in the area of sleep neurobiology, that an important event or important component of the transition from wake to sleep involves activation of these GABAergic, and in the case of VLPO neurons, also GABAergic neurons in the preoptic area that send descending projections to several of the key ascending arousal systems. And that through the very powerful effects of monosynaptic GABAergic inhibition, a relatively localized cluster of sleep-active neurons in the preoptic area is able to help promote a generalized and integrated suppression of activity in several key arousal systems, and hence hasten the transition from waking to sleep. So the electrophysiological data I've showed you indicate that these cells become active at the transition from waking to sleep, and perhaps they are a very sort of short near-term event during those transitions, sort of helping flip the switch, in a sense, from waking to sleep. And so while it's true that wake-sleep transitions can be very sudden and switch-like in certain circumstances, there are also these slower time course aspects to the regulation of sleep, which Dr. Kaskadin spoke of this morning, and which are diagrammed here in the two-process model from Borbet, where there is a hypothetical process S, which increases its strength, increases the tendency to sleep as a consequence of prior waking, and then is dissipated during sleep, and of course interacts with the circadian oscillator. So we ask the question then, what is the relationship of the discharge or the activity of these preoptic sleep regulatory neurons with respect to these slower time course aspects of sleep regulation modeled by process S? So what we did was to record neurons in both the MNPO and the VLPO in rats, and to try to follow individual neurons across a period of baseline sleep, a period of sleep deprivation, and a period of recovery sleep, to see if the discharge or neuronal activity was correlated with what we could determine to be increasing homeostatic sleep pressure. This was work done by Akhtab Alam in the group, which has recently been published online in the Journal of Neurophysiology. So this is the basic experimental paradigm. So what you're looking here is essentially a 5-hour continuous recording, or we're recording unit activity, EEG and EMG. This is a baseline period. This is a 2-hour period of sleep deprivation induced by gentle handling. That is, whenever the animal starts to show evidence of falling asleep, the experimenter intervenes, taps on the cage, if necessary, strokes the fur of the rat with a cotton swab to keep it awake. And then after 2 hours, the animal is released to recovery sleep. See, we have these little tick marks or dots here in this top trace. Those are little event markers that indicate every time that the experimenter had to intervene to keep the animal awake. So initially, when sleep deprivation starts, it's relatively easy to keep the rat awake. The interventions are only required every 10, 15 minutes or so. But at the end of 2 hours, the animal becomes much more sleepy, and that's reflected in these increased frequency of attempts to fall asleep. So this is sort of a nice behavioral measure of how sleepy the animal is while it's awake. So what you can see here, this is a neuron that is sleep active. There's not much here in the baseline, but this is a period of wakefulness where the activity is low. Here's a period of sleep. When the sleep deprivation starts, the discharge of the cell is very low. But as sleep deprivation continues and the animal becomes progressively sleepier, the waking discharge of this neuron increases dramatically, such at the end of 2 hours of sleep deprivation, the waking discharge rate is higher than the baseline discharge rate during sleep. And when the animal is released into recovery sleep, there is initially maintenance of elevated discharge, but then as recovery sleep progresses and sleep debt presumably is resolved, or homeostatic sleep pressure is resolved, the discharge rate of the cell declines. This just shows higher time sleep periods from here, B, waking early in the sleep deprivation period, C, waking late in the sleep deprivation period, showing the dramatic change in discharge rate of the cell. So these are mean data for 11 MNPO neurons studied in exactly the paradigm that I just showed you. This is during sleep deprivation, and this is waking discharge rate, now expressed as a percent change from baseline waking, during each of the three 30-minute quartiles of the sleep deprivation period. This is a plot of the number of sleep attempts, our behavioral measure of sleepiness. And there's clearly a significant increase in the waking discharge of these neurons as a group during the second 2 hours of the sleep deprivation period, corresponding to this increase in the behavioral measure of homeostatic sleep pressure. This is during recovery sleep. This is now non-REM sleep discharge rates. What you can see is that initially, during the initial recovery sleep periods, the discharge of the cell is significantly elevated over baseline sleep rates, as is EEG slow wave activity, the sort of electrophysiological biomarker of sleep pressure. And then as recovery sleep progresses in association with a decline in EEG slow wave activity during sleep, non-REM sleep discharge returns to baseline levels. So this indicates that these cells then function very much like they are sensitive to or responding to something like process S. They are becoming activated in proportion to prior time awake and in proportion to evidence of increased sleep pressure. And then that process dissipates during recovery sleep, and that is also reflected in the discharge of the neurons. So we did similar kinds of experiments in neurons located in the ventrolateral preoptic area. I won't show you the data in the interest of time, just to say that they're by and large similar. VLPL neurons show this increase in waking discharge rate during sleep deprivation, not quite as dramatically as cells in the MNPO, and they also show an increase in discharge rate during recovery sleep. I have a bit to talk about some REM-related cells, but I think I will skip that in the interest of time, just to point out that the findings that we indicated with these changes with respect to sleep deprivation were unique to the sleep-active neuronal population. So we also studied a group of wake REM-active neurons and a group of state-indifferent neurons located in the lateral preoptic area of the rat, and these did not show any systematic changes with respect to sleep deprivation and recovery sleep. So in this area of the brain, at least, where we found the sleep-active neurons that were responsive to homeostatic sleep pressure changes, they were localized to the median preoptic nucleus and the ventrolateral preoptic nucleus. So I will skip this about the REM-active cells. It's an interesting story, but not quite as clear-cut. So we're continuing to study these cells and perhaps have a clearer story in the future. So the question then becomes, what's the mechanism that underlies these the effects of sleep deprivation, recovery sleep on the discharge of these neurons? Is there a neurochemical signal or neurochemical signals that might explain the changes we see with sleep deprivation? So given the nature of process S and the aspects of sleep that it was designed to model, that is, the pressure accumulates gradually during waking and dissipates with some slow time course during sleep, it's often been thought that some sort of the production of some sort of endogenous sleep factor or factors and their metabolism during sleep could underlie these slow time course effects. And one of the major candidates, and probably the endogenous sleep factor that has the most compelling evidence for a role in sleep homeostasis, is adenosine. So adenosine is produced as a byproduct of ATP metabolism, so it reflects sort of energy levels of energy use in cells. In the brain, adenosine can be produced from ATP either extracellularly or intracellularly, and there are adenosine transporters that can move adenosine in and out of cells. Most important for understanding what the key sources of adenosine might be with respect to sleep regulation, in the brain, adenosine can be produced both in neurons and in astrocytes. And one of the things that can activate ATP release and extracellular adenosine production based on activity in astrocytes is increased glutamate release, which tends to be high in waking. There have been studies showing that if you disrupt the ability of astrocytes to release ATP and therefore lead to increased adenosine production, you can disrupt sleep. So it's important to remember that the adenosine that might be involved in sleep is not necessarily sourced solely from neurons, but probably also a very important source is astrocytes. So of the many kinds of evidence implicating adenosine in sleep homeostasis, there are data like these that show that these top two panels, A and B, are studies done in cats from Bob McCarley's lab in collaboration with Tarja Porkka-Heiskanen, shown here initially during spontaneous sleep in the cat that adenosine levels tend to be higher during wake and lower during sleep. Here there's a sleep deprivation paradigm where cats are subjected to 6 hours of sleep, and these are studies involving microdialysis sampling of extracellular fluid, followed by HPC analysis for adenosine. The dialysis probes here are placed in the basal forevein, very close to the lateral preoptic area, and you can see that sleep deprivation results in an increase in adenosine release, and that recovery sleep is associated with a decrease. These are similar findings in adult rats. Again, with a prolonged period of sleep deprivation, there's an increase in adenosine that then plateaus and decreases in recovery sleep. You also can see changes in the cortex at a somewhat slower time course and not quite as dramatically in terms of the magnitude. So what do we understand about the mechanisms through which adenosine can influence sleep? There are two main adenosine receptors in the brain. The A1 adenosine receptor is an inhibitory receptor. It's located in many brain areas where it has universally inhibitory effects. So the potential sleep-promoting effects of adenosine would be acting through the A1 receptor to inhibit several of the key arousal systems that we talked about earlier, cholinergic and non-cholinergic neurons in the basal forebrain, hypocretin-orexin neurons. A1 adenosine receptors also exert inhibitory effects on thalamic and cortical neurons, which promotes EEG synchrony. The A2 adenosine receptor is excitatory, has a much less restricted distribution in the brain. Its highest density is located in the striatum by far. But there have been some evidence from Haishi's group and Tom Scanlon's group at Harvard that adenosine may have excitatory effects on VLPO neurons acting via the A2A receptor. So we specifically asked the question, does the A2A receptor, is the A2A receptor involved in activation of the specific sleep-active GABAergic neuronal populations in the MMPO and VLPO? It's essentially a pharmacological study where we infuse an A2A receptor agonist and look at CFOS expression in GABAergic neurons in the two nuclei. We infused the drug via two routes. One was ICV into the lateral ventricle, and the second route was into this subarachnoid space, just rostral to the basal forebrain, rostral to the optic chiasm. So the rationale for doing this second infusion site was that, the goal was to put a cannula directly here in front of the septum and into the subarachnoid space just in front of the optic chiasm, and to deliver the drugs directly into the subarachnoid space. So this is, in fact, the reason for doing this is a very effective way to deliver drug to the median preoptic nucleus, which sits right here, just adjacent to the subarachnoid CSF behind the septum. And the ventrolateral preoptic area sits at the very base of the brain here, so in immediate contact with the subarachnoid CSF in the chiasmatic cistern located here. And work from Haishi's group has shown that microinfusions of the endogenous thaumagen prostaglandin D2 are most effective when placed in this portion of the subarachnoid space. So this demonstrates the effects of drug infusion, again an A2A receptor agonist, into the lateral ventricle versus the subarachnoid space. In the lateral ventricle we use 2 doses, 4 microgram and 12 microgram. In the subarachnoid space we use a single dose, and this just shows that the higher dose in the ICV route induces a suppression of waking and an increase in non-REM and REM sleep. The lower dose, as we had predicted, would be much more effective in stimulating the critical sleep regulatory areas, and the low dose, here the 4 microgram, produces a decrease in waking, increase in non-REM and REM sleep. So we then look at the CFOS expression in GABAergic neurons, in the MNPO and VLPO, here in the rostral and caudal components, here in the Vilpos core and the extended VLPO. And there is an increase in FOS expression in all the areas at the high dose via the ICV route, and an increase with the lower dose via the subarachnoid space route. This last condition here is an important control condition because the drug infusion induces sleep. So if you see an increase in FOS expression in the sleep-active neurons, it's difficult to interpret whether that's just another example of the neurons being active when there's an increase in sleep amount. So what we did here was to diffuse the drug and then keep the animals awake during the 2 hours post-infusion before the sacrifice. And what you can see here is that even though the animals are not permitted sleep, there's still an increase in FOS expression in the GABAergic neuronal population in MNPO and VLPO. So it indicates that the drug can activate the cells in a sleep-independent manner. A second experiment we did was to take a A2A receptor antagonist now, so a drug that blocks the effects of adenosine excuse me, at the A2A receptor, and infuse it during a period of sleep deprivation. So the paradigm was we did a 3-hour sleep deprivation period starting near ZT2, 2 hours after lights on. During the last 2 hours, we infused this A2A antagonist, permitted the animals 2 hours of recovery sleep, and then killed and harvested brain tissue. So this demonstrates that there's a dose-dependent increase in waking, a dose-dependent decrease in non-REM sleep during recovery sleep, during this 2-hour recovery period, as a result of this drug infusion being delivered during sleep deprivation. And this is the FOS cell counts, where there is dose-dependent decreases in both MNPO and VLPO. So this suggests that the presence of the A2A receptor antagonist during the sleep deprivation period diminishes the homeostatic response during recovery sleep. So we essentially then decided to try to walk that finding back to our single unit studies to determine if the A2A, in a more direct manner, whether A2A receptor activation was important in driving the homeostatic related changes in neuronal activity in the VLPO neurons. And for this we used the method that we have used before, where we combine our microwire bundles to record neuronal activity with a dialysis probe placed adjacent to the recording electrodes, which we can then deliver drug through, sort of a reverse dialysis. So we can actually record neurons in a baseline state, push CSF through the cerebrospinal fluid through the dialysis probe, and then at some point switch to drug and deliver drug to an area around the dialysis probe that will include the areas where we're recording neuronal activity. So we can look for sort of the acute effects of a local drug delivery on neuronal discharge. So we did that with our paradigm of this, sleep deprivation and neuronal recording. So this is the experiment that I had shown you earlier, where we have a baseline period identifying a sleep-active neuron. We do a 2-hour period of sleep deprivation. You can see in the second half of the sleep deprivation, the waking discharge of the cell increases. And then at this point, just before we release the animal to recovery sleep, we deliver an infusion of the A2A agonist, a local infusion of the A2A agonist for 15 minutes. And you can see here that the drug effect results in a near immediate suppression of this waking discharge, which returns very close to baseline levels from the elevated level that were present during the sleep deprivation before the drug, and also suppresses the non-REM related discharge of the cell during recovery sleep. Eventually, when the drug washes out, baseline sleep activity returns. This is just, again, a higher time sleep at B and C, B and C, showing the difference, the drug effect on non-REM activity here versus here. And this is mean data for groups of cells. So for a population of 6 sleep-active neurons recorded in the ventrolateral preoptic area, studied in this way, infusion of the A2A antagonist suppressed both waking discharge and non-REM sleep discharge. Significantly, there was no effect of drug on a small population of awake-active neurons. So this then is more direct evidence that what is driving these wake-induced changes or increases in the activity, the sleep regulatory neurons in the preoptic area, is activation mediated via A2A receptors. Okay, for the last 10 minutes or so, I want to change gears just a little bit and talk about sleep development and how these sleep regulatory circuits that we've described in adults might be involved in the normal maturation of sleep homeostasis and sleep regulation in the infant. Again, these are studies that were done in rats. So there's a bit known about the development of sleep homeostasis in rat and to a lesser extent in mice. And one thing that's clear is that not all aspects of the homeostatic response mature at the same time. So very basic aspects of the sleep homeostasis are present in the period immediately after birth. So within 1 or 2 days of birth, if you deprive an infant rat of rest, because there's no EEG differentiation of sleep at that age, but you deprive it of rest periods, you see a clear rebound in increases in resting activity or in active sleep-like responses when you discontinue deprivation. So that very basic aspect of sleep homeostasis, an increase in rest in response to rest deprivation, is present very early. The sleep-wake differentiation of the EEG in the rat emerges around 10 to 12 days. There might be a little bit of controversy about that. It's not something that's been extensively studied. The sort of what we think of as the key biomarker of homeostatic sleep pressure in an adult, that is increased EEG slow wave activity, that doesn't occur to somewhere around postnatal day 20 to postnatal day 24 in the rat. And also about that time you start to see evidence of sleep consolidation in response to sleep deprivation. So we were then interested in trying to understand what is the role, of any, of sort of maturation or changes in the activity of these sleep regulatory neurons and sleep regulatory circuits that we've characterized in adults to the development of homeostasis in sleep homeostasis in rats, and in particular measures of activity of NMPO and BLPO neurons. And we focus on the fourth postnatal week, P21, P30. And since things are reasonably well characterized up until around P20, but it's not clear how much and how dramatically things change during that fourth postnatal week. So this is an example of our initial study was done by Irma Gavilia from our group, where we're looking at here, this is essentially a repeated measures design where she studied the same rats. Here studied during a baseline period on postnatal day 21, here after 2 hours of recovery sleep on postnatal day 22. All right, and then here we have the same rat studied during baseline on postnatal day 29, here postnatal day 30. So what you can see here is that if you look at the baseline sleep compared to recovery sleep following deprivation, you can see that there is in fact an EEG response. You can see EEG slow wave increase here versus here. There is an increase in sleep amount as well, but there's actually no change in the degree that sleep is consolidated. That is, there's no change in sleep bout durations during recovery sleep. Whereas in the rat at P30, again you see the increase in EEG slow wave activity. The scales are different, so there's a sort of further maturation of the EEG delta power expression during sleep. But there's also a significant increase in the duration of bout length in recovery sleep. Maybe it's a little hard to see in this figure here, but here's the mean data shown in table form. So here we have spontaneous sleep on P21, recovery sleep on P22, spontaneous sleep P29, recovery sleep on P30. So this demonstrates that you do see the basic homeostatic response of a decrease in waking and increase in sleep at the earlier stage. You see no change in non-REM sleep bout duration, whereas there's a significant increase in bout duration in the older rats. You can also compare the absolute values of EEG power, and you can see the further maturation of the EEG slow wave generation system. These are CFOS GAD cell counts in the median preoptic nucleus. For P22 rats shown in the open bars, P30 rats in the closed bars during conditions of spontaneous sleep, sleep deprivation without an opportunity for recovery sleep. So the animals are sacrificed right at the end of the sleep deprivation period. And here, recovery sleep following a 2-hour period of sleep deprivation. So what you see in P22 rats is there is an increase, a significant increase in FOS-GAD cell counts in the sleep deprivation period versus the spontaneous sleep period, which is somewhat similar to what you see in the adult. But there are dramatic changes in FOS expression in the NNPO by P30 compared to P22. So what you see here is that sleep deprivation without recovery sleep is higher than spontaneous sleep, and recovery sleep is intermediate between the two. So this is the pattern, the exact same conditions and outcome measures in the adult rat, and you can see that by P30, there's been a dramatic maturation of the responsiveness of these NNPO GABAergic neurons to changes in homeostatic sleep pressure. So it really identifies this fourth postnatal week as a sort of a sensitive period for the maturation of these cells in sleep regulatory circuits. Here are the data for the ventrolateral preoptic area, and this is in a sense more dramatic in that at P22 there's actually no effect of any condition on FOS-GAD cell counts in the VLPO. And by P30 you start to see the beginnings of an adult-like response, where you see an increase in, most clear here in the extended VLPO, a decrease with respect to spontaneous sleep as a result of sleep deprivation, and an increase of activation during recovery sleep. So just some recent, this is now unpublished data, but we've been sort of trying to look at patterns of unit recording of neuronal activity in these developing rats. In part because the neuronal activity gives you a sort of a better temporal resolution of issues related to state transition and dynamic changes in sleep pressure than the CFOS method does, because the weakness of the CFOS method is the slow time course of the synthesis of the protein and then the protein's degradation. So really a technical achievement that was accomplished by Irma Gavilia and Natalia Sansova in the group of being able to achieve stable neuronal recordings in rats as young as, actually they were successful in rats as young as P19, but this is an example from a P21 rat showing unit recordings in the median preoptic nucleus during natural sleep and wakefulness. So the first thing they looked at was simply state-dependent discharge in the two age groups, P22 and P30. So this is active wake, quiet wake, non-REM and REM sleep. So again, as was indicated by the FOSS, the median preoptic neurons are sensitive to state at P22, and you can see there's clear increases from active wake to quiet wake to non-REM sleep. Here the REM sleep amounts, like we see in the adult, are the same. But at P30, there's a significant, I would say, maturation towards the adult pattern in that the cells are, the mean discharge rate increases, and they're more proportionately active during sleep, during waking at P30 than they were at P22. So they also did the sleep deprivation paradigm. Recording baseline data to identify the cell, and then doing a 2-hour sleep deprivation period. So here again you have this now expresses discharge rate during the three mean data for the 4 30-minute quartiles of 2-hour sleep deprivation period. And what you can see here in the P22 rat is there's no significant change across the 2 hours of sleep deprivation, no significant increase in discharge rate, whereas there is a significant increase during the last 2 to 3 quartiles of the deprivation period in the older rats. And the pattern in the P30 rats much more resembles what we see in adult rats. So further evidence that there's maturation of these homeostatic systems expressed at the level of neuronal discharge in the sleep regulatory neurons occurring during this 4th postnatal week. So the last thing I'll show you is an attempt our first attempt to try to integrate the potential role of adenosine signaling in these developmental changes. And so what we did in collaboration with Radhika Bashir and Anna Kalinchuk at Harvard University in the Boston VA system was to measure extracellular levels of adenosine in the lateral preoptic area of P22 versus P30 rats, and do that in the context of sleep deprivation to determine is there a difference in the ability of either the neuronal systems or the astrocytes in this part of the hypothalamus to generate adenosine, to produce adenosine in response to sustained wakefulness. And these are the data. So what we have here is baseline, 3 hours of sleep deprivation, 2 hours of recovery sleep. So these are extracellular adenosine levels, again assessed with microdialysis and HPLC. Everything is expressed as a percentage of baseline. In the P22 animal, there's absolutely no effect or no change in adenosine levels across the conditions of sleep deprivation and recovery sleep, whereas the P30 rats exhibit the adult pattern of increased extracellular adenosine in response to sleep deprivation and then decreases during recovery sleep. So this suggests that part of the developmental changes or maturational changes that are going on are the ability of neurons and astrocytes in this part of the brain to either alter the metabolism in such a way to affect changes in adenosine levels. So just then to summarize, what I've tried to convince you of is that, and I think there's reasonable agreement in the field, that activation of these GABAergic neurons in the preoptic area are sort of important near-term steps in the switching from waking to sleep. The ability of these relatively localized clusters of GABAergic neurons to promote global changes in arousal is by virtue of their descending projections to critical ascending arousal systems. But in addition to playing a role in sort of near-term sleep switching, there's also evidence now that these neurons are responsive to sort of slower time course stimuli or signals that are associated with increasing sleep pressure as a result of sustained wakefulness. And activation of the VLPO and MNPO neurons via adenosine A2A receptors appears to be an important component of the homeostatic response to sleep loss, and it's showing you some preliminary data that maturation of these sleep circuits and adenosine signaling in the hypothalamus and the basal forebrain are potential factors in the development of homeostatic sleep regulation, and also identifies the A2A adenosine receptor as a target, potential therapeutic target for addressing sleep disturbance in both adults and perhaps during development. So again, thank you for the invitation to attend the Congress and for your conversation.
Let's get started with the Q&A session. Are there any questions for Dr. Zsymusiak?
So that was an excellent talk. Thank you for sharing. Do you think that, so that adenosine, I assume you're saying it's coming from neuronal activity, and that's where the ATP breaks down and adenosine accumulates, right? So what happens when people go into REM, well, animals go into REM sleep, because the brain neuronal activity goes up again? Does that produce more adenosine, or how does that?
So the issue of REM sleep is sort of it's a dirty little secret of the adenosine hypothesis, because you would expect that at least several of the cell types in the hypothalamus and lateral hypothalamus will become activated during REM sleep. Again, the one possibility is that the neurons are only one source of adenosine, that the astrocytes have been recently identified as a second and potentially important source. So it's sort of unknown how that REM, non-REM dynamic would affect adenosine release from the astrocytes. There might be a slower time course effects of adenosine, in fact related to intracellular signaling changes, so that the short-term changes in neuronal activity that occur during at least the relatively brief REM episodes in the rat wouldn't impact the general level of activity of the sleep regulatory circuits. But it's certainly an unanswered question, and it's one that hasn't been addressed, and is difficult to address with the BOSS method, because REM episodes in rats and mice are very brief, and sleep in general is fragmented. So it's a possibility we can start to get an understanding of some of these REM, non-REM related changes with using the unit recording approach, which gives you a much finer temporal resolution.
Any other questions?
Thank you very much for your excellent conference, and I will speak in Spanish.
I have the question of what happens. With the inducing effect of adenosine in the case of sleep induction. Is this mediated by a cholinergic tone, or what would be the relationship with acetylcholine, if there is any, and the frequency of neuronal firing in preoptic neurons for sleep induction?
Excellent question. So my response would be that the immediate transition from waking to sleep really involves sort of the final step. It involves the increased neuronal discharge of the GABAergic neurons, which then cause a final level or the threshold level of suppression in several of the arousal systems, including the cholinergic system. The way I see changes in extracellular adenosine levels acting is sort of to reset the system, to balance the system more towards sleep and towards wake, so that you're going to have A1 adenosine-mediated inhibition of the cholinergic neurons, so you're going to have reduced cholinergic tone, and you're going to have some activation of the sleep system. So this is sort of the example, I think, of what we're experiencing during the extreme sleepiness while we're awake as a result of sleep deprivation. You have adenosine causing decreased cholinergic tone, increased GABAergic activity of the sleep regulatory system, so the overall level of vigilance is reduced, and the ability to tip the system from wake to sleep is balanced in favor of sleep. So I definitely see that there's a role for sort of tonically reduced cholinergic tone, but that's sort of the slower time course effect of the adenosine. But then the near-term event is the final burst of firing from the sleep-active neurons, which then puts the brain to sleep.
Oscar.
Sorry. Thank you very much, Ron. Wouldn't you expect to see more activity in the neurons of the preoptic medial and ventrolateral area in the youngest animals, because they have more slow waves in general, no?
Right. Well, so I think that that's an interesting question, and at some point you might expect to see a change, but it's a question of where we are in the sequence. So it's like Mary Carskadden said this morning that 10-year-olds are the ideal sleepers because they have the most consolidated deep sleep that occurs at the optimum circadian time. So it's a little hard to equate developmental stage in a rat to a human, but you know, just for background, rats wean at about postnatal day 17, and they're considered to be adults at around postnatal day 60. So I sort of see this fourth postnatal week as there's sort of still an increase in consolidation and an increased circadian organization of sleep. But at some point you would expect to see perhaps even like at P40, P50, they might even show increased activation with respect to adults, and then it should eventually decline. So that's how I would interpret it.
Una última pregunta. Muy bien. Thank you very much.
Thank you.
Zsymusiak.